Hello everyone. My name is Shuo, and my presentation today is about accurate high-throughput cryptic binding site prediction using protein language model. The proposed model called LampSite can identify cryptic binding sites from protein sequence without 3D protein structures as input. Before introducing the model in detail, I would like to introduce about the background and motivation of our work. And the task we are investigating is related to ligand binding site prediction. And binding site serves as a key interface where proteins engage in fundamental cellular processes, making it an attractable target for drug molecules. For example, here shows ion channels that can open in response to the binding of a ligand, which play an important role in the treatment of several diseases. And given the importance of binding sites, identifying ligand binding sites of protein is an essential step in the pipeline of drug discovery. With the information from the identified ligand binding sites, different downstream processes can be done. For example, the ligand binding sites can provide the bounding box as the surface region of molecular docking. They can also serve as surrounding structures of ligands for structure-based drug design. Besides, they provide necessary information for structure-based work screening. And for small molecule ligands, the binding sites also located in exposed concave pockets. However, some binding sites only appear to be pockets in the presence of ligands. Are there still any echoes? Hmm. Yeah, I think from my side, I cannot do any other modifications. Okay, maybe I just continue. And such binding sites are called as cryptic binding sites. For example, the apple structure shown at left doesn't have exposed concave pockets for the ligand to bind. However, the ligand can change the conformation of the protein and exposes a concave pocket between the two alpha helices for binding, so that the cryptic binding sites enable the targeting of undruggable proteins that lack exposed pockets in the apple structures. To identify ligand binding sites or cryptic binding sites, many computational methods have been proposed. However, we find them all have certain limitations. Regarding the existing computational methods for binding site prediction, we find that top-performed methods predominantly rely on the availability of ligand bound and rigid 3D protein structures. Given such limitation, these methods will face challenges when trying to identify binding sites for newly sequenced proteins without experimental 3D structure data. Even with computationally predicted 3D structure, for example, from AlphaFold2, people find that experimentally determined binding sites can be obstructed by low confidence regions or wrongly predicted side chains, thus affect the performance of binding site identification. Besides, for proteins with high precision hollow structures available, detecting without those hollow structures available, detecting cryptic binding sites can be challenging for such models. Regarding the existing computational methods for cryptic binding site prediction, we find that most of them require data or information from MD simulations to provide information on the opening events of cryptic binding sites, which are resource intensive. In recent months, There have been new generative methods proposed for directly predicting 3D binding complex structure between a pair of the ligand or protein. With the predicted complex structure, potential cryptic binding sites can be reviewed. However, such generative models are diffusion-based, which are time-consuming and thus cannot be easily adopted in high-throughput compound screening. To address the aforementioned limitations, we set the following goals for cryptic binding site prediction. We seek to avoid the dependence on rigid 3D protein structures and diffusion-based generative modeling, as well as the IMD simulation data, while incorporating the dynamic nature of cryptic binding sites accurately and efficiently. Um, how about now? Still echo? Let me continue. To achieve this, we are inspired by the power of protein language models, which can be used to obtain protein information without the need of 3D protein structure as input. And in detail, we use the ESM2 pre-trained protein language model. In the original ESM2 work, by using the attention maps in the pre-trained language model, it is found that protein contact map can be predicted unsupervisedly, which demonstrates that ESM2 can implicitly capture the 3D structure of a protein. We further assume the predicted contact map can be used to jointly model the apple and hollow structures of each protein. As shown in these figures, the conformational chain between the apple structures in gray and the hollow structures in blue that lead to cryptic pocket formation do not significantly break the folding pattern of a protein. Based on this, we propose our language model, power cryptic binding site predictor, or called LAMP-site, that incorporates ESM2 computing embeddings and ligand information for cryptic binding site prediction. The overall architecture is shown like this, and I will introduce the modules one by one. First of all, the protein feature extraction. We actually take the protein sequences as the initial input to compute protein representations. With a frozen pre-trained ESM2, we compare protein. We compute a protein contact map and protein residue embeddings for each protein. And our model also takes the input of ligand information, which is to predict ligand-specific cryptic binding sites. For ligand feature extraction, we use the 2D ligand molecular graphs or SMILE strings to be fed into a frozen pre-trained UniMo, which is an SE3 transformer-based framework for universal molecular representation learning. And the pre-trained UniMo can generate item-level embeddings for each ligand. Besides, we use RDK to utilize a 3D conformer for each ligand to compute the corresponding ligand distance map. Based on the residue embeddings and ligand item embeddings, we compute the interaction embedding for the protein ligand pair, which models the interactions between protein residues and ligand items. For each pair of the residue embedding from the protein and item embedding from the ligand, we concatenate them to form the interaction embedding. Furthermore, we update the interaction embedding by taking the protein contact map and ligand distance map as geometric constraints. To accomplish this, we use a variant of the evil former block in AlphaFold 2 as our interaction module, which contain a similar two-step operations, including the triangle multiplicative update and triangle self-retention. Different with the operations in dual former that all modes are from protein, we assign them to be either protein residues or ligand items to have the computations. Then we found the previous interaction embeddings together and use a pooling over the dimension related to the ligand items to predict a score for each residue. A higher score indicates a stronger interaction between a residue and ligand, meaning that the residue likely constitutes part of the binding site. For the training process, our model is optimized to minimize the binary cross-entropy loss between the predicted residue scores and the ground truth labels. For cryptic binding site level prediction, we further filter out residues with low scores and cluster the remaining ones to form binding site based on the contact map information. To compare our model with 3D-based methods, we map the predicted scores to the corresponding residues in the 3D protein structure. After introducing the model architecture, we would like to talk about the experiments and results. In our experiments, we used the ICPDB 2017 database as our training set, which contains residue level labels from about 20,000 binding sites. For the evaluation of cryptic binding site prediction, we used the same validation and test set constructed by Pocket Manner, which contains residue level labels that specifically indicate the belongings of residues in cryptid binding sites. Regarding the metrics and baselines in our experiments, we first use residue level metrics, including the PR-AUC and ROC-AUC, since the labels in the dataset are directly residue level ones. The baselines for comparison include CryptoSite and Pocket Manner, which are two typical deep learning-based methods that all require information from MD simulations. Besides, to have comparison with 3D-based method or generative models, we use 3D level metrics, which are DCA and DCA success rate. DCA denotes the distance from the center of the predicted binding site to the closest ligand highway item. And the baseline operation include P2 rank, which is a typical method for general binding site prediction that takes the rigid 3D protein structure as input. Here we fit couple structures to P2 rank for cryptid binding site prediction. A neuroplexer and dynamic bind are two diffusion-based generative models that predict ligand-protein binding complex. Based on the predicted 3D binding complex, we denote the residues to have a distance smaller 7 angstrom to the closest ligand highway item as binding site residues. For comparison on residue level metrics, LAMP-site outperforms the baselines on both metrics. And our model has the following advantages. Okay. First, the help of implicit 3D structure information captured by protein language model. LAMP-site doesn't require 3D protein structures as input. And second, LAMP-site is not specifically trained on cryptid binding site related data or explicit MD simulations that capture the dynamic nature. And for the comparison on 3D level metrics, we listed the detailed DCA values of each ligand protein pair in the test set that contained the cryptid binding site. By comparing the DCA success rate with a threshold of 4 angstrom, which is a commonly used value in binding site prediction, we find our model outperforms the baselines. Besides, while all other baselines have more than 15 angstrom DCA values for certain pairs, our model's largest DCA is only about 7 angstrom on the test set. This success rate plot for different DCA threshold also shows the power of our model. Especially for the 3D-based model P2 rank that takes the Apple structures as input, it performs worse on all DCA thresholds. And our model exhibits competitive performance when compared to the generative models. Furthermore, we compare the models regarding the inference speed on the same device. We find that our model achieves more than 100-fold speed up when compared to the diffusion-based generative models, while having competitive prediction performance. This implied the promising power of LAMP-side when adopted in high-throughput compound screening. And to evaluate the power of using LAMP-side predicted residue scores in downstream tasks, we performed experiments on binding site affinity prediction. And the proposed model architecture involves a frozen pre-trained LAMP-side module that can provide residue scores that identify binding sites. Then we use the same way as in LAMP-side to extract features from protein and ligand, and the extracted embeddings are multiplied with the LAMP-side predicted scores as constraints. And finally, these embeddings are pooled and concatenated for the prediction of binding affinity with a multi-layer perception. For the dataset, we use Campbell 31 and the random scaffold split for the comparison with baselines. And the results show that our model, even without LAMP-side, can outperform the baselines on both metrics. And with the predicted scores from LAMP-side, the performance can be further boosted. Since we freeze the LAMP-side part in fine-tuning, it will now introduce extra computational burden to the model. And the inference speed for the binding side, for the binding affinity prediction is very fast. And while our model has demonstrated good performance, it is important to acknowledge several limitations to be solved in the future. Due to memory constraints in self-attention, our model is now trained on relatively long protein sequences. And besides, our model takes a single chain as the input. Its performance in multi-chain scenarios may be limited. Lastly, we will work on evaluating the model's ability when incorporating in more downstream tasks. Thank you so much.
I don't know if you have questions online, but we have this one here.
Hi, really nice work. I was wondering, two questions. One of them is sort of short, the other one not so long. First is why not use structure if in the end these are predictions you're making in the structure, so you're mapping them back to the structure. And then the second one is when you compare with the other methods, for example, feature rank, I know in most cases it will make more than one prediction. So are you considering all the predictions, the top n+2, or just top one prediction? Thank you.
So thank you for your question. For the first one, we here map the predicted scores back to the structure is just used to have some visualizations and compute the 3D based metric. So we didn't use the structure here because we just want to make our model to be efficient, and in certain scenarios using the computational predicted structure is will have some effect for the bad effect for the binding side prediction due to the low confidence region predictions and some side chains that would affect the binding side. And besides, for cryptic binding site prediction, in many cases, only the Apple structure is available. So we still want to heavily rely on the protein sequence information. And for the second question, we for the P2 rank setting, we just use the top n setting. So when there are n binding sites in the complex, we just select the top n predicted binding region for the prediction. Actually, we find the improvements using a larger n value doesn't have significant improvement on the final prediction because the input structure we are using are just Apple structures. And in many cases, the cryptic binding sites, they are still not exposed on the surface, so that it will be not feasible for P2 rank to do predictions for those reasons.